All right, Dr. O again. Here's a Unit 4 exam review. This one will be slightly different. I like to put the human in human biology, so I have a degree in human biology, and I like to focus. Most of the resources that I've created are focused on the human organ systems. So, which chapter 16 is the last one we're covering. Then we start to get into things like ecosystems and populations and evolution and stuff. And I'm well trained in those things, but since most of my classes are anatomy and physiology classes and not these human biology classes that also incorporate these other things, I don't have as many resources. So for chapter 17 and 19 in this unit, I'll be defining some things that you won't have seen in any resources that I've made for you. So I do a good job, or as good a job as I can, of finding really good resources for you. So like for example, I have already put a couple videos about Punnett squares and how we use them to do these crosses and different things in your resources for chapter 17 that I didn't make. So I do personally, I try to make all my own resources. I think it's very important for the connection that I develop with you guys, but don't teach a lot of human biology classes. If I continue to teach this every year or every semester, then I'll certainly will make more resources myself. But just so you know, that's why this unit and the last unit, you will see a lot more resources that I've curated for you rather than produced. But reproductive system, I hit really hard with my own stuff. So let's jump in. Chapter 16. Basic differences between male and female reproductive system. So the big thing is the male system's job is to make and deliver sperm. Very simple. The female system is much more complex because, so seriously, the male gonads are like always make testosterone, always make sperm, have a way to get rid of it. The female system has to make gametes, has to control female sex hormones, but also has to be a home for that fertilized egg, that zygote, once that occurs. So the home for the developing embryo and fetus is a big addition to the system. And then on top of that, you have breast milk production. So men have to make gametes and get rid of them. Females do the same thing, but also have to nurture that baby as it's developing for 9 months and then feed it with breast milk production. So much more complex. The other big difference is, I'm not saying, so men certainly have peaks and valleys in testosterone levels, and they change over your lifetime too, but the female system, the timing is critically important. If a male's like always make testosterone, always make sperm, always get rid of sperm or whatever, that's pretty simple. The female system, it's all about timing because, during a 28-day cycle, there's a very short window where you can get pregnant. And that only works if the two cycles, the uterine and ovarian cycles, work at the same time. Or a time together. You can produce the world's greatest egg and you can ovulate, but if the uterus isn't ready for it when it gets there, well, you're out of luck. And then vice versa. You could have the best uterus in the world, but not ovulate correctly or at the right time and you're in trouble. So way more functions in the female system, and also that timing is so important. So sperm versus egg, they're your gametes, the sex cells. So they're haploid, meaning they only have one set of chromosomes, and we need two sets or two pairs of chromosomes to be a human, which is why 23 chromosomes from a sperm, 23 from an egg makes that human. But the other key difference is we churn out sperm constantly. Like every day, a male can make 300, 400 million sperm, whatever. And during meiosis, when one cell becomes four, all four are functional sperm. With the egg, you only need one egg at a time. It's all about quality with eggs and quantity with sperm. So you're only going to have one functional egg, and those other cells become what are called polar bodies, and they're recycled. So those are some major differences there. The male system, so again, it's all about producing sperm and moving it. So the male system, you've got inside the scrotum, you have the testicles. So into the testicles, you're going to have the cells that produce testosterone and also the cells that cause the maturation of sperm, like your nurse cells, and the testosterone cells are called interstitial cells. But also, inside the testicles, you have the seminiferous tubules. That's going to be where sperm are being produced and matured. From there, we go to what's called the ductus deferens. You may have heard it called the vas deferens before, or maybe you've heard the term vasectomy. Then that's going to connect to the short little ejaculatory duct, and then we have the urethra. The accessory structures, the big ones would be the seminal vesicles. They produce about 70% of semen. So semen is only 5% sperm or spermatozoa, 95% of these extra secretions. So the big one is not the prostate gland. You might think that, but because you've heard of it. But the seminal vesicles or seminal glands produce about 70% of semen. They also produce the food for sperm, which is fructose primarily. The prostate glands are important, but the prostate gland, we're not even sure exactly what the prostate gland does. I mean, we know it produces an antibiotic called seminal plasmin and a few other minor things, but the main reason when everyone knows the prostate gland is because the urethra runs through it. It's kind of a bad design flaw. If the prostate gland gets enlarged because of cancer or just hypertrophy or an infection, it pinches the urethra and makes urination difficult. So the prostate gland is famous, or infamous, but it's not super important. And then then we have the bulbourethral glands. Their job is to lubricate the penis and also try to help neutralize any acidity, because one key difference between male and female reproductive tracts is the urethra is a shared tube in men. We use the same tube for urination as we do for reproduction, whereas in a female, the urethra is separate from the vagina. They're close to each other, but they're separate. All right, sperm production. Let's see, external genitalia. You've got the testicles inside the scrotum, and you've got the penis in men. All right, sperm production, temperature, and fuel. I already mentioned with sperm production that you're constantly churning out sperm. Every one of these sperm stem cells can become 4 functional sperm, whereas it's only going to be 1 functional egg. Temperature, that's an important thing. Like, why are the testicles outside the body? Like, if I could play God, if I could design a human, I would have the testicles encased in bone because they're so pain sensitive. Well, the reason they are is because pain is what protects them, inhibiting from doing things that are hurting them. But they have to be outside the body because sperm function at a temperature about 2 degrees Fahrenheit cooler than the rest of the body. So the reason the testicles are outside the body and the reason they move is to maintain their temperature. If it's cold, the testicles will climb up towards the body to stay warm because of muscle contraction. And if it's warm, they'll drop away from the body to stay cool. So temperature matters a lot with sperm. Then the primary fuel I already mentioned is fructose. The seminal vesicles produce fructose, and that is part of semen. The male hormones, pretty straightforward. You've got, I already mentioned this in the last unit, but follicle-stimulating hormone is what tells the gonads to make mature sperm. Luteinizing hormone tells the testicles to make testosterone. So testosterone is the key male sex hormone or androgen. Female system, the actual reproductive tract is probably less complicated, but everything else is more so. So you've got the gonads. When you think gonads, you might think testicles, but the gonads are where gametes are produced and sex hormones. So the gonads in males are the testicles, but in females are the ovaries. So you got the ovaries. Then you have the oviduct, which the oviduct is the term for the tube where the passage of the oocyte or egg occurs. But in mammals and us, it's going to be called usually you hear it called the fallopian tube or the uterine tube. Then you have the uterus, which is going to be where the fertilized egg would implant or embed and where you would nurture that growing human. And then you have the vagina, which is the tube that's used for sexual reproduction and for labor and for delivery. All right, so we talked about what an oviduct is, and in us it's called the fallopian. It's actually, the technical term now is uterine tube, but fallopian tube is still the most common term. So the menstrual cycle is the ovarian plus the uterine cycle. I mentioned before that the two are intimately connected and they have to be timed together. So there's a real good image, and I did a video on this, but so you can call the whole thing the menstrual cycle if you want. But the ovarian cycle is, the cycle should be 28 days long. So the first half is going to be dominated by follicle-stimulating hormone and estrogen. And that's going to cause the development and growth of the follicle, which is where the egg is. Then you have ovulation, which the next question there. What hormone surge causes ovulation? A surge in luteinizing hormone is actually what causes ovulation. That egg is going to erupt from the follicle, and now it's going to be heading towards, trying to find sperm, basically. Which is why the other half, the uterine cycle, has to be tied to this, because during the follicular phase of the ovarian cycle, you have menstruation, the menstrual phase, where you get rid of last month's uterine lining, then the proliferative phase, where you build this month's uterine lining. And then if that happens, so the uterine lining should be waiting for a fertilized egg when ovulation occurs. Then the second half of the cycle, once the egg has been released, is now going to be dominated by the hormone progesterone, and that's going to cause the secretory phase of the uterine cycle, which means that it's going to get thicker, glands are going to be secreting, it's waiting for a fertilized egg. So it's way more complex than this, make sure you watch that video. But all right, so when is a woman most likely to get pregnant in a typical 28-day cycle? Right about day 14 there in the middle. or give or take, because day 14 is when ovulation occurs. That egg is now going to start heading down the oviduct, heading down the uterine tube, and somewhere in that uterine tube is where egg and sperm should meet if it's going to happen. Then that fertilized egg, which would be called a zygote, will start dividing, and then when it gets to the uterus, it'll be ready to implant and embed, and that's when you start to see the placenta take over, and the placenta basically becomes an endocrine organ that keeps your sex hormone levels where they should be while you're pregnant. So, all right, let's see. Well, let's see. I lost track of where I'm at. I'm sorry. So when we're right there in the middle, right there, give or take around day 14, in a typical 28-day cycle will be when pregnancy would be most common. So they generally say there's like a 2 to 3 day window, but you can certainly probably get pregnant on 7 days. That's why the rhythm method or birth control, which only uses not having sex during key times around ovulation, is pretty ineffective, because that window is probably a lot larger in some people. All right, what hormones look for in a home pregnancy test? So one of the cool things that happens here is after ovulation, the corpus luteum, like where the follicle, where the egg was, that starts to release progesterone. But then if you get a fertilized egg, this shell around the fertilized egg is actually going to start to release HCG, human chorionic gonadotropin. So a home pregnancy test is a urine test looking for HCG in your urine. So and if the levels are high enough, then chances are you're pregnant. Where does fertilization usually occur, and where must the fertilized egg end up for pregnancy to occur? So I already mentioned this. Fertilization usually occurs in the oviduct, in the fallopian tube. Most people would say about 2/3 of the way down the path. So the eggs head in one way, the sperm climbs up the other way, and they meet in the fallopian tube there. But then where the egg must end up, it must embed itself in the uterus in order for pregnancy to work well. All right, here at next I just have the birth control methods. There's so many of them, so I'm just going to kind of scan through what the book says about this, because I don't have a video on birth control methods. So abstinence, of course, it's 100% effective. If you don't have sex, you can't get pregnant. So then we have surgical sterilization. So in a man, it's called a vasectomy, where you actually cut and seal both ends of the vas deferens or ductus deferens. Then you have a tubal ligation in a female, which actually does the same thing with the oviduct. As a man, I've had a vasectomy, whatever, but I think it's, when in doubt, the man should be the one to do it. It's a much, like the guy that did mine, he's done 1,000 of them and never had any serious repercussions. It's definitely a more aggressive, potentially dangerous surgery in a female. But then there's tons of other birth control methods. You have like IUDs. They can either be what they can do is actually just irritate the uterus that keeps fertilization or implantation from occurring. Some of them actually have chemicals in them as well, so they can be like a chemical and physical form of birth control. You've got injections and contraceptive patches and birth control pills. They all influence hormone levels. You have condoms and diaphragms and cervical caps. These are the kind of things there's female condoms as well. These are the kind of things that can just kind of catch sperm and keep it from getting to where it would need to get. There are spermicides that actually can kill sperm, these kind of things. So tons of different examples. So I already mentioned cervical caps and diaphragms already. So, now I just mentioned that rhythm method, which is basically periodic abstinence, like knowing your cycle, knowing when you're most likely to get pregnant, and then abstinence then. So there's abstinence all the time, or just abstinence during certain periods. That I think withdrawal would fall into the same there, where you withdraw the penis before ejaculation. But we know, like, if you follow the rhythm method or the withdrawal method, there's about a 1 in 4 chance you're still going to get pregnant, over any given time. So. All right, so on the flip side, infertility. So infertility is the most common cause of infertility in the United States, and it can be men too. Sperm counts are dropping in men, number of functional sperm is dropping in men, so men certainly bear a lot of this burden, but the most common cause of infertility in the United States is called PCOS, polycystic ovarian syndrome, which is actually, it appears to be to deal with insulin resistance. It affects hormone levels. So it is certainly preventable and treatable, but that is a very common cause of infertility. If you're going to do things to enhance fertility, there's lots of minor things you can do, but you have in vitro fertilization and other big things. But I wanted to break that down into ZIFT and GIFT as two major methods of enhancing fertility. So they're both types of in vitro fertilization, but GIFT is gamete intrafallopian transfer. ZIFT is zygote interfallopian transfer. So with GIFT, you're putting sperm and egg in the uterine tube, in the oviduct, where they would become a gamete. With GIFT, you've already had the egg fertilized and then deposit that. So just kind of cool. All right, then you've got with STDs. There's tons of them here in the book. The most common. And there are some, like I said, I used to actually work, part of my job as a designated civil surgeon was to monitor syphilis coming into the U.S. Of course, we got HIV and different types of hepatitis and stuff. But right now, the most common STD in America is considered to be chlamydia. And then lastly here, what kind of cancer is the HPV virus associated with? Actually, quite a bit. I mean, you've got it causes genital warts, but the big thing is the cervical cancer. So that's number one. But my wife works in gastroenterology, and there are people that develop like rectal and anal cancer from HPV, from anal sex, and these kind of things too. But the big one is HPV virus. Think cervical cancer. All right, we're going to move on to chapter 17 then. Okay, so chapter 17 is about cell reproduction and differentiation. So cell reproduction would be how cells divide to become multiple cells. Differentiation is how cells become the type of cells they're going to be, which makes them different from other cells. So the cell cycle is going to basically be a combination of interphase and then the mitotic phase. So interphase is the time in the cell life cycle where the cell isn't dividing, but it's preparing for division. So this is going to be when your DNA is duplicated, these types of things. But the cells, for most cells, 90% of the time they're in interphase, which means the cell isn't focusing on division, it's doing its job. You're going to live 80, 90 years, and you're only going to be focusing on reproduction for a handful of those years. Well, same thing with cells. So during interphase, the cell's just doing its job. It's not dividing, but it's preparing to divide. So some cells never leave interphase. Cells that don't ever divide, they never leave interphase. Other cells, like stem cells that are constantly churning out new cells, they're barely ever in interphase. But I like to say that most cells spend about 90% of the cell life cycle in interphase. So interphase would be called the S phase. When yes, the cell might be growing, but it's not dividing, but it's definitely preparing for division. So interphase has that S phase where the DNA is duplicated, and then the G2 phase where the cell is preparing for cell division, but it is still growing. Then you have mitotic phase. So the word mitotic phase is the division of the nucleus. So when you think of mitosis, you often think of the division of body cells, but technically mitosis requires the rest of these processes. So the mitotic phase or M phase is when the nucleus divides, and then a cell can divide. One cell becomes two. Then you're in the G1 phase where you have the cells growing like crazy. The S phase is when the DNA is duplicating for the next time. So it goes G1, S, G2, and then the mitotic phase. So just think interphase, the cell's growing, it's doing its job, preparing for division. Then you have the mitotic phase, which involves the 4 steps of mitosis: prophase, metaphase, anaphase, and telophase, or telophase. And then cytokinesis is actual cell division, this one cell splitting into two. Okay? All right, differentiation, I already told you that's when a cell becomes its own kind of cell, which makes it different than other cells. I'm not going to go through all the details of transcription and translation here. That would take an hour. But DNA structure and DNA replication. So remember, DNA is double-stranded, has a sugar / phosphate backbone, and it's made of the 4 nucleotides A, C, G, and T. Replication is called a semi-conservative process, which means that when you have one DNA strand divides and becomes 2, each new daughter DNA molecule is made of an old strand and a new strand. So we're not making brand new DNA; we're making copies basically. So that's how one strand becomes 2. When DNA is built, it just follows the complementary base pairs. Wherever you see an A, it lays down a T. Wherever you see a C, it lays down a G. So those are your complementary base pairs. A pairs with T, which means T pairs with A, and C pairs with G. All right, mutations and DNA repair. So a mutation, we always think of it as a bad term, but mutations are any change in DNA. So most mutations are bad because we're the evolutionary winners. We're here because we're good, right? Which means that overall our DNA and genes are good. So most mutations are not going to help, but some will. So A mutation is just a change in DNA. Now, thankfully, we don't see a lot of mutations in us because we have a lot of DNA repair mechanisms. I mean, dozens of them. The enzyme DNA polymerase can actually proofread as it's going. We have light repair, excision repair enzymes, you name it. So most mutations that occur in the human body will be fixed, which is great. But occasionally mutations are going to lead to positive, beneficial mutations. I like to think of it as a book. If you look at a book, there's almost always a typo in a book. Maybe an extra space, maybe the word "the" 2 times in a row, maybe a misspelled word. Well, if you were to close your eyes and randomly point somewhere in that book, chances are you didn't make it better. But it is possible. So think of the lottery. Every mutation, I talk about this a lot in microbiology, every mutation would be like playing the lottery. So usually you're going to lose, but every once in a while you're going to win. So, and how that works is mutations lead to alleles. Alleles are something that's a bigger deal in the next chapter, but alleles are like different copies, different versions of the same genes. Like, for example, in humans, hair color, eye color, blood type, these are all alleles. It's all human blood types and human hair and human skin color, etc., but different versions of those genes. And then which versions of those genes become more common? That's evolution. So without mutation, there wouldn't be any differences. And without differences, we couldn't have evolution. We couldn't have winners and we couldn't have losers. So, all right, the details of transcription versus translation, I'm not going to go through here, but transcription is the conversion of DNA into RNA, whereas translation is the conversion of RNA into proteins. Let me kind of help you with that. So a transcriptionist changes the format of something, doesn't change the language. So DNA and RNA are both in the nucleic acid language, but they're in different forms. DNA is double-stranded, whereas RNA is single-stranded. DNA has the sugar deoxyribose in its backbone. RNA has the sugar ribose in its backbone. DNA is made of A, C, G, and T. RNA is made of A, C, G, and U. So it's only changing the form, but not the language. So like when I used to use a medical transcriptionist, I would give them little tape recorders, little tapes, little cassettes, which, it's all digital now, but I actually have like 7 digital recorders I use, and they're probably all full right now because I never have time to get them emptied. But so I give them to a transcriptionist, these little cassettes, and she'd give me back these documents, these reports. So she didn't change the language. If I said something wrong, it's still wrong. She just changed the form. Translation, now translators change languages. So I work with translators too. When I was a designated civil surgeon, I would say a sentence in English and then my translator would say it in, whether it's male or female, would say it in Spanish. So translation changes the language. So translation is turning RNA, which speaks the nucleic acid language, into proteins, which speak the amino acid language, right? So that's how I remember those two terms. Think about what a transcriptionist does and a translator does. Okay, your genetic code and codons. So the genetic code's really cool. All living things share the same DNA. Whether it's the grass under your feet or a cow or you, a bacteria doesn't matter. We're all made of different versions of variations on A, C, G, and T. So genetic code is the language, the blueprint for building all living things. Codons are the 3-letter sequences or triplets of bases that we use to build all the proteins that exist. So the codons are how we turn RNA into proteins. You read these 3-letter sequences and convert them into amino acids. So I won't go through all the details, the videos cover it, but the 3 phases of transcription and translation, so they're different and have different players involved, but both transcription and translation are three-step processes. That's initiation, something starts it. With transcription, it's called the promoter sequence of DNA. With translation, it's called the start codon, or AUG. Then you have elongation, just the process is occurring. With transcription, you're building RNA. With translation, you're building a protein chain. Then we have termination in step 3. With transcription, it's called the terminator sequence of DNA that stops the process. With translation, it's called a stop codon or a nonsense codon. So they use the same 3 stages or phases, just different terminology. All right, the steps of mitosis, I'll try to keep this as simple as possible. During prophase, the nucleus disappears. We don't actually know why. So during prophase, it's prophase, metaphase, anaphase, and telophase, or PMAT is the steps of mitosis. Prophase, nucleus disappears. Metaphase, the chromosomes line up at the equator, or the center of the cell. Anaphase, those chromosomes, which have already been duplicated earlier during interphase, are going to be pulled into 2 piles. Then during telophase, 2 new nuclei form. Then after telophase is over, we have cytokinesis, which is actual cell division. The cell splits in two. So I like to separate cytokinesis from mitosis, but most books don't. So those are the steps of mitosis. Watch the video on that. Mitosis versus meiosis, they're pretty similar. Mitosis is, you're going to see one round of cell division. Meiosis, you're going to see 2 rounds. So in mitosis, one diploid cell, or one cell with 2 sets of chromosomes, is going to divide into 2 diploid cells, 2 cells with 2 sets of chromosomes. With meiosis, you have 2 rounds of cell division, so that one cell ends up becoming 4 cells, but they're haploid. They only have 1 set of chromosomes. That's why, meiosis only produces sex cells. So in humans, we have the sperm and the egg. They only have 23 chromosomes. All other human cells have 46. So your body cells with 46 chromosomes are diploid or diploid. Your sex cells are haploid. So that's the main differences. So basically, if you look at meiosis, it has prophase, metaphase, anaphase, and telophase. Then it basically does it again. Prophase 2, metaphase 2, anaphase 2, and telophase 2. So you basically have 2 complete cell cycles like you'd have in mitosis, and you just don't have interphase a second time. So that's one way to look at it. All right. Then the book goes into some stuff about cloning and different types of cloning, and we already mentioned differentiation, but I do want to clear, I'm not going to get into the philosophical arguments about cloning, but I would say that if you're, everyone's going to draw a line somewhere differently. Some people will say no cloning, but I think you have to understand there are 2 major differences. There's reproductive cloning and therapeutic cloning. So reproductive cloning will allow you to clone an organism, which we know is happening. There's been examples of that happening. I can't say whether a human's ever been cloned, but that's what reproductive cloning is. So like Dottie the sheep would be the first example of a mammal that was cloned. They can clone cows to get beef from them and things like that. But therapeutic cloning is different. Therapeutic cloning, you're creating cells, you're creating maybe tissues or someday whole organs. They're designed to treat diseases. So to me, reproductive cloning is let's say, do you want to clone a human? Well, I would say, what's the point in that? Because there's already too many humans, and there are so many children that need good homes. Why would you clone a baby when you could take care of a kid that's already alive? That's my opinion. Therapeutic cloning is different. If someone has, right now, if someone has a problem with their heart, they'll have to do a heart transplant, which is far from perfect because you have to basically shut off the immune system, or else the heart's going to be killed by your immune system. So my dad had an aortic valve replaced. So the rest of his heart is 70 years old, and he has this aortic valve that came from a cow. Well, someday the hope is that therapeutic cloning will say, you know what, if we're going to go in anyways, let's give you a 20-year-old's heart, your own, right? So you don't have to worry about transplant drugs. You don't have to worry about the immune system rejecting it. Like, we're just going to give you a younger version of your heart. That's pretty cool. But before we get there, we're talking about things like creating a heart patch. So after a heart attack, they cut out that diseased portion of the heart and just patch it, something like that. So we're not there yet, but it's pretty cool. So reproductive cloning, think producing a whole new organism. Therapeutic cloning is all about treating disease. Okay, chapter 19, last chapter in this unit. This unit only has 3 chapters, so I think there's 3 chapters in the last unit as well. I'm trying to remember. So chapter 19, what are genes? So a gene is a sequence of DNA that codes for a functional product. So in most organisms, most of DNA isn't genes. So only about 2% of our genetic material is actually from our genome is a gene. So genes are sequences of DNA that code for a functional product. Now, you might hear somebody say codes for a protein. Most functional products are proteins, but not all of them. So that's a better, more specific definition. All right, several more terms here. Genotype versus phenotype. So your genotype is all the genes that you have, right? Your genetic makeup. Your phenotype would be what are called your observable traits, which genes are being expressed. And then it's important you note they're not the same thing. So your genotype is all your genes. Your phenotype is the ones you're using. So think of it this way. We all have recessive genes that are being hidden, that are being dominated by dominant genes. So those aren't being used. We all carry genes that we're not using that we can pass on to our offspring. So if you're carrying a disease you don't have and you can give it to your children, that's a perfect example of your genotype not being the same thing as your phenotype. Another important terms we'll talk about here, especially with the Punnett squares and the like, we have the terms homozygous versus heterozygous. So homozygous means you have two copies, two of the same kind of copies of a gene. Heterozygous means you have two different ones. So you can be homozygous dominant and have two dominant copies of a gene. You can be homozygous recessive and have two dominant copies of a recessive gene. And you can be heterozygous, one of each. So that's just an example. If we're talking cystic fibrosis, that's a recessive disorder, which means you'd have to be, the only people that have the disease are homozygous recessive. They got a copy of the bad gene from mom and dad. So if you have the disease, you're homozygous recessive. If you're homozygous dominant, you not only don't have the disease, you're not even carrying it. If you're heterozygous, you have one good copy of the gene and one bad copy, which means you don't have the disease because that dominant gene is hiding the recessive gene, but you carry it. So that's these terms homozygous and heterozygous mean. All right, so what is a Punnett square and why is it useful? So a Punnett square is the simplest tool we have to represent like inheritance patterns, but the main reason I like it is it allows you to predict the probability that a certain genotype will be inherited. So like, what are the odds that mom or dad are going to pass certain gene combinations on to you? Now, first we have to understand that probability has no memory. So when we say like, so if you use a Punnett square, and hopefully you watch the Punnett square videos, if you have, if mom and dad are both carriers of cystic fibrosis, well, that means there's so we'll say it's like the book, big A, little a. So big A is the good dominant gene, little a is the bad one. So if you use the Punnett square and you see that both mom and dad are carriers, they're going to have a capital A and a small a. Well, that means that there's a 25% chance that you're going to get two capital A's and you're going to be homozygous dominant, not only not have the disease, but not even carry it. That's 1 in 4 chance, 25%. There's a 2 in 4 chance, so two of these squares in a Punnett square are going to be capital A, lowercase a. So you still don't have the disease because you have to have two bad copies to get it, but you're going to be carriers. And there's a 25% chance that you're going to get lowercase a, and you now have cystic fibrosis. But you have to understand that this mom and dad could have 17 kids in a row, and all 17 of them could have cystic fibrosis, or none of them could have it. Because it's just like rolling dice. The dice don't care what you rolled previously. These are just basic probabilities. So you could have 3 kids in a row that have cystic fibrosis, or 3 kids in a row that don't. Just so you understand that. So, but that is why we use it. This would have been what Gregor Mendel, the father of genetics. He didn't even know what a gene was, but he's considered the father of genetics. And maybe you've heard of Mendelian genetics or Mendelian inheritance. But he figured this stuff out without knowing genes. He just knew that things were being transmitted from parent to offspring. He was able to figure this stuff out. So the Punnett square is how we can figure out the odds of these things happening. So, speaking of Mendel, let's look at a couple of these laws. Let's talk about. We already talked about these, but dominant versus recessive alleles or traits. So I already mentioned this. A dominant gene is going to dominate or hide a recessive gene. So, most diseases are recessive genes, so you have to get 2 bad copies. But there are dominant genes that can be bad too. I'm not saying good or bad, but the crazy thing to me is you can't look at a gene and say, "Oh, that's the dominant one." For some reason, dominant genes dominate recessive genes, but we can't actually say for sure. Why? So there are some dominant gene conditions. So if you only have 1 copy of the gene, you will have that trait or that condition. But the recessive ones, you'd have to get 2 copies unless we're talking about sex-linked traits, which I'll get to in a little bit. So let's look at these 2 laws, the law of segregation and the law of independent assortment. The law of segregation, as simple as I can put it, says this: mom and dad both have 2 copies of a gene, right? They might be different copies, but they both have 2 copies or 2 alleles, 2 copies of a gene. Well, each of them only contributes 1. So that's why a Punnett square works. You're going to get one of mom's 2 genes, 2 copies, and one of dad's 2 copies. And they're going to fight it out, whichever one's dominant or recessive, and that's going to determine how you turn out. So if you think about you, first of all, I like to say you're not a combination of your 2 parents, you're a combination of your 4 grandparents, but because, the sperm that came from dad, egg that came from mom, basically you've got like 23,500 genes. You got 23,500 coin flips. Is this gene going to come from mom's mom or mom's dad? Is it in the egg? Is this gene going to come from dad's mom or dad's dad in the sperm? And that's going to determine, what sperm and egg made you. But then the law of segregation says that you're only going to get 1 allele from each parent. So and then which one you get is completely random. Which egg gets fertilized by which sperm. So it's very random, and that's good. We'll look at why randomness matters in a little bit. So that's the law of segregation. Next, we have the law of independent assortment.
So this is important when you look at these two trait crosses. We'll get to in a moment. When we're looking, well, we can't just do a simple Punnett square and have, where we just have a single trait being looked at. A two trait cross is where you're going to look at multiple traits. But so what this shows is that the law of independent assortment says that whether or not, it's like in the book, it talks about E's and W's. Whatever E you got from mom, whatever E you got from dad is completely independent and doesn't care about what W you got from mom and W you got from dad. So every gene, that's why I talk about these coin flips, every gene is just this random assortment that you get from mom and dad. So now, of course, there are now some of the processes that lead to this randomization, there are genes that sit close to each other. So, of course, they're more likely to be passed on together. Like a good example would be genes for red hair and genes for freckles are really close together. So oftentimes you see people with red hair that also have freckles. But you don't have to, right? You can have red hair and not have freckles, or vice versa, of course. But so that's what the law of independent assortment says, is that the individual genes don't care what other ones you got from mom and or dad. So the two trait crosses, just again, you can look in the book at that and the videos that I put together for you. But if you're going to look at more than one trait at a time, you can't just do a basic Punnett square. You have to do these more complex. You can look at two traits. Now you can make these as huge as you want, but they get more and more complicated. All right, this next series of words are going to be connected. We have complete dominance, incomplete dominance, and codominance. So if we have a complete dominant situation, it is a dominant gene that completely dominates a recessive gene. If we haven't, so that means that you might have a second copy of that gene, but it's not being utilized. The dominant gene has dominated the recessive one. Incomplete dominance means that you have genes that can't completely dominate other genes, right? So there's like in flowers. Let's say you have a red flowering plant and a white flowering plant. Their offspring will be pink in some situations because the red can't completely dominate the white can't completely dominate the red. So that's why you get that variation in between the two. So that happens in humans as well. I think that in the book they talk about, you have a straight hair gene and a curly hair gene. Neither can dominate each other completely. So that's why you'd have a wavy hair pattern. So these would be genes that don't follow that complete dominant versus recessive, and there's going to be an intermediate between the two. So that's what incomplete dominance is. Then the next one we have is codominance. So I gave two good examples here, both blood type and sickle cell. Codominance means that you're going to have multiple dominant alleles that can dominate something, but what you can have where both alleles are going to be expressed equally. So let me give you an example. Yeah, the ABO blood typing system. So you have type A blood, B, AB, and O. Well, O is recessive. The only way to have type O blood is if you got a type O blood gene from mom and dad, because you have to be homozygous recessive. If you got an A and an O gene, you're going to have type A. If you got a B and an O gene, you're going to have type B. So both the A gene and B gene completely dominate O and hide it. But A and B cannot dominate each other. So if you get a type A gene from mom and a type B gene from dad, your blood type will be AB. So that's an example of codominance. So the sickle cell example, the reason this one is so important is because this is actually why in some parts of the world, the sickle cell anemia still exists. So this is a really good example when you're talking about genetics, because when you look at is a sickle cell gene a good or bad thing, whenever you're talking from an evolutionary biology standpoint, the answer is depends. It depends on the environment. So in the United States, there's no reason to want any of the bad, if you want to call them, copies of the sickle cell gene. But in Southeast Asia and sub-Saharan Africa, where malaria is still a major problem, you actually want to be heterozygous. So you want one quote-unquote bad copy. So with codominance, so let's say if you have two normal hemoglobin genes, that's exactly what you want in most parts of the world. But if you have that in these places where malaria is still a major problem, malaria kills 1 million people a year. I mean, the malaria is the reason that the mosquito is the most dangerous animal on the planet. It basically goes mosquitoes number 1 and humans are number 2. So in parts of the world where you're not going to get malaria, then you want 2 good copies of the hemoglobin gene. So you don't want the sickle cell anemia or the sickle cell trait. But even in parts of the world where malaria is a problem, if you get 2 quote-unquote bad copies, then you're going to have sickle cell anemia, and that's not good either. So because anemia is a bad thing. So you want to be homozygous for the normal hemoglobin in the United States and parts of the world, malaria is not a problem. You never want to be homozygous for sickle cell anemia. But in those parts of the world where malaria is a problem, you actually want to be heterozygous. You want one copy of each. So you want one gene for normal hemoglobin and one gene for sickle hemoglobin. And the reason for that is because it actually, you're still going to get plenty of oxygen, but it decreases the risk that you're going to get malaria. But the main issue is, if you're less likely to get malaria, you're less likely to get a massive infection. But the main thing is you're less likely to get cerebral malaria, which is the fatal kind of malaria, mainly. So actually, here's an example of where having now. Now, if you have sickle cell anemia and you live in sub-Saharan Africa, you're still protected, but you also have sickle cell anemia. So this heterozygous situation, this codominance example, where both genes are can't dominate each other, can actually protect you. So that's a pretty cool example there. All right, next, what do crossing over, independent assortment, and random fertilization do? So crossing over is the fact that, during meiosis, when you get each individual chromosome, you're going to get, there's basically four, you can get, for chromosome 7 or whatever, you can get all of your mom's mom or all of your mom's dad's, or you get a mixture that's mainly mom's mom or mainly mom's dad. That's this crossing over process that occurs during meiosis. In the department with independent assortment, we already talked about the fact that which genes you get from your mom and dad are random, which is why I like to say you're a combination of your 4 grandparents. Then random fertilization, which sperm wins to get with which egg, and it's not the fastest. It actually, usually it's maybe 100 or 300 sperm in that gets there, whatever. But which sperm and which egg made you? Like, just think of how many genetic combinations your parents could make, and the fact that they made you, makes you, think about how many options for humans there are that will never exist. So the fact that you exist means you're a pretty big deal. But what do these things do? They allow for diversity. Like, you and your siblings are closely related, but you're wildly different as well. And that's the cool thing. So, now these are kind of mathematical games, but in order for 2 people to have 2 copies of the same kid, not counting, basically like, twins or, identical twins are basically clones, but not counting that, a mom and a dad would have to have 10 to the 2100th children. That's not 2100 kids. That's a 10 with 2100 zeros behind it. That's how many kids. That's how many different genetic combinations will occur before you have the same kid twice, basically. Of course, that'll never happen. That's an ungodly number. But when you're talking statistics, anything over 10 to the 30th isn't even possible. So we're far beyond that. The record for children between a man and a woman is 69. I think she was from Hungary. She was pregnant basically every year she could be, and she had twins, triplets, and quadruplets almost every time she had kids. But the record for a man is over 700. I think it's 732. But he was a sultan and had a harem. So 1 man, 1 woman, the record is 69. So we're talking about this massive number. So I just mean is you're unique. There's never been another human being like you. The exact same genetic combination as you. No, it's never happened. So that's pretty cool. All right. Nature versus nurture. So we're talking about all this genetic stuff. Like, you can leave a class like this thinking that genetics are hugely important, which they are, of course. But I'm really big on the nurture side, especially because of this whole idea of epigenetics. So nature is your genetic code, right? Here's what I like to say about your genetic code. Your genes load the gun. The environment, the nurture, pulls the trigger. So you can have a high risk of certain diseases, but if you do all the right lifestyle things, you're going to greatly reduce those chances. I also like to say that your genes, they predispose things, they don't predetermine things. So you can be hardwired for stress, but you can find ways to deal with it. You can be hardwired to have a really short temper, but you can find ways to deal with these things. So I'm a huge fan of nurture and the environment. That's why I take my job as a dad so seriously. The environment is a major, major player. So yes, nature matters, but now we have this whole idea of epigenetics, which means, to me, it means nurture matters even more. Even if you thought it was 50-50 before, now we know that the environment your genes are in influence how they work. So that's called epigenetics, EPI genetics. And it's actually, so the environment can influence how your genes operate. So epigenetics is, it can't change your DNA. It can't change the A's, C's, G's, and T's that make you, but it can turn genes on and turn them off. It can speed them up and slow them down, things like that. So personally, I'm definitely more on the nurture side of that spectrum. All right, single gene traits versus polygenic traits. Just so you understand, single gene traits are, a single gene determines something. Can you roll your tongue or not? Do you have hair on your knuckles or not? Those would be simple examples of single gene traits. Some types of muscular dystrophy, sickle cell anemia, and cystic fibrosis. Those are examples of single genes that can influence health or disease. But most traits are going to be polygenic, meaning that it takes more genes than one, and some way more than one. You're going to see a lot more variation. So things where there's wild variation between humans are usually polygenic. It's not on or off. It's a continuum. So think about like height. The example the book used, I think, was height. So there's really short people and really tall people and people in the middle. Well, that's because height, there isn't a height gene. There's not a small gene and a short gene. If there was, we'd just be really small or really short. There's this wild variation. And the environment plays a role too. So your genes, if you have tall genes, and however many there are, if you have tall genes but you don't eat well and you're malnourished, you're not going to get tall. So intelligence, heart disease, people believe that no one even knows, but there could be 1,000 genes that influence intelligence. So when you look at like cloning and trying to create, trying to genetically modify and engineer humans, there's not a smart gene. There's probably 1,000 of them. There's probably 2,000 genes that influence your heart disease risk. These are examples of polygenic traits, which is another reason why we have so much variation between humans. All right, 2 more and we're done. The genotype and environment alter the phenotype. So certainly your phenotype is the observable traits that come from your genes. And so your genes matter, don't get me wrong, but genes load the gun, environment pulls the trigger. So if you have the two worst copies of the BRCA genes, which are the breast cancer risk genes, they're BRCA genes they're called, then you might have an 80% chance of getting breast cancer, but you don't have a 100% chance of getting breast cancer, which means if you do all the right things and avoid things that speed up mutation and do things that protect you from mutation, etc., you may be able to win that fight. Now, those genes are really setting you up for failure. That trigger is going to be pretty sensitive, but the environment matters, and it matters a ton, right? So I want to make sure you understand that. I don't like to think that our genes really predetermine anything. They increase or decrease your risk of things, absolutely. And you could have the best breast cancer genes imaginable and still get breast cancer because you do all the wrong things, right? So the environment do all the wrong things. I said, do all the wrong things, not wrong things right. Sorry about that. Then lastly, your sex-linked traits. So any genes, any traits that come from genes on the X and Y chromosomes are going to be sex-linked. The reason I want to bring these up is because here's an example of where one copy of a recessive gene can cause something to happen. So in women, that's never the case, or not never, but in a normal woman, they're going to be XX, 2 X chromosomes. There are women that are XO. They got an X but didn't get another chromosome. That's called Turner syndrome. But so in a normal woman, if you have a bad copy of a gene on the one X chromosome, you have a second X chromosome to protect you. So you would need 2 bad copies. So like if a woman's going to be colorblind, so you're going to see that your sex-linked traits are always way more common in men. Like usually 9 out of 10 are going to be in men. So for a woman to be colorblind, she would have to get a colorblind gene from mom and dad because both mom and dad gave her an X chromosome. So there is a second gene that can dominate a recessive trait. So mom is XX with 2 X chromosomes. So mom truly does have 2 pairs of each chromosome. Men have 2 sets because men are XY. So normal men are XY. So if dad got a colorblind gene from mom. So his mom wouldn't have to be colorblind, but she was carrying it. If a man receives not dad and mom, but if a man receives a colorblind gene from their mom on the X chromosome, there is no second X chromosome to hide it. So a man can be colorblind with 1 colorblind gene. A woman would need 2. So same thing with like hemochromatosis, which is an iron storage disease, probably the most common genetic disease in the United States. These are examples of, so a man is the only one that can get a recessive condition with 1 copy. So if a man gets a bad gene from on an X chromosome from his mother, there is no second X chromosome to protect it. So that basically means that your sex-linked traits are more likely to affect. So like sex-linked disorders are way more likely to affect men, but they got the disease from their mom and they would pass it to their daughters. Because if I'm colorblind as a male that's XY, the colorblind gene is on my X chromosome. I can pass that gene on to my daughter because she'll be XX, an X from me and an X from the woman, my wife. But I can't pass it on to my son. So I can't make my son colorblind because if I give my son my X chromosome, he's not my son because you're always going to get an X from mom. If the dad gives an X, it's XX, it's a female. So these sex-linked disorders or traits are passed back and forth between males and females. Mom passes the sex-linked trait usually onto a boy who then passes it to his daughter and vice versa. That's why they say things skip generations sometimes. So that's why sex-linked traits are important, because, and they're more likely to affect men, these negative ones, because there isn't that second chromosome to protect it. All right, crush this test. See you in Unit